So we're just going to get into the next part of this protein basics lecture, and this will cover the idea about protein folding and misfolding and how this can then lead to pathology.
Right, so I just put a real simple intron exon there, and those sequences, those genes can be mutated, and if those genes are mutated, right, and so this is now where we can start to make a little bit more of the next phase, right, is you have your mRNA, and we're just going to go right to the exons, right, the expressed parts, those are E's. Okay, so that would lead to a mutation in the message RNA that's pumped out of the nucleus. And then the ribosomes would read that incorrectly, right? So if we have some sort of mutation that's carried over from DNA, that's carried over into mRNA, and then the protein that comes out is going to have a different amino acid sequence or altered amino acid sequence, and then that will affect its folding properties, and that will affect its function. Okay, so if we take now this mRNA and go to the normal route, right? Once we have if we have our amino acid sequences coming out in the correct order, I'll just make a couple of weird whatever sequences, different shapes, right? The hydrophobic, or the polar, the non-polar amino acids, the positive and negative charges, as they come out of the ribosome, they will start to fold in a particular configuration. Right, so you go from we're going to be talking about this today, the primary to the secondary, which is these alpha helices, these beta sheets, right? It's just these different folded structures that start to take form. I mean, they're connected. And then really to where I think is the most important function of the protein is that primary or the tertiary structure. Okay, so that's where those particular amino acids, and I'll try to do it with the colors, right? You have the orange, we have this purple, and I'll make those the alpha helices, and some of the greens, and then I don't know, whatever. Okay, so you have that tertiary structure that really is now how that protein folds its native conformation, and it goes through conformational changes. Okay, and the important thing to keep in mind about these proteins, so I'll just say that this is our native conformation. It's the lowest energy, and it's where the protein can kind of sit in its natural environment. So there was a question, of course she's not here, but that's all right, whether or not you could have put proteins into different environments, and probably not. I mean, I said that you could, so let's say you have a water-liking protein that folds naturally in water and puts its non-polar amino acids in the inside, so it has a very low energy structure. If you were to put that into a fat-liking environment, it would tend to fold. It would probably fold correctly with its non-polar amino R sequences on the outside, but it would be in a higher energy state, and it would not be very well off. So this is where, the protein folds in its particular environment based on its amino acid sequence where it has the lowest energy, so it's native conformation. And you can move that protein in and out. So it will go now through these conformational changes, and this is kind of where we start today. So you have a conformational change, I'm going to draw that out a little bit longer, where the protein goes to a different folded configuration. Okay, so you have the orange maybe, and maybe this middle sequence straightens out. And then it can do a particular function. And that's just absolutely perfectly fine, right? So it goes into a conformation change as long as it comes back, okay, to its native conformation. So it bounces in between these different folded states, and there's different ways in which it can do that. Okay, so the last part of this lecture are the post-translational modifications, which are typically these reversible modifications of proteins that can lead it to this conformational change, and then they disappear and it goes back to its native conformation. What we're going to be talking about today is when the protein should be in its native conformation, but it essentially gets stuck into this abnormally folded or misfolded configuration. So we could still have the same protein, right? But now maybe it's folded in this different aspect for whatever reason. I'll just put this here, right? But it's stuck there. It doesn't go back. And that's where you get into problems. So what you see is that with a protein that's folded in the proper configuration, you have these positive and negative charge interactions, right? Those amino acid are groups that are positively or negatively charged. So they will, as long as they're in the right place, they will fit together. The hydrophobic, hydrophilic interactions, right? So shown in red. So all of those amino acids have come out in the right sequence, in the right place, and it allows that protein then to form its native conformation. If things are out of sequence, and so they just show one actually, that's thrown off, if a mutation changes one of the charged amino acids, that part of the protein becomes less stable, and I mean it says wobbles around. I mean, so really what that means is that it's in a higher energy state and it's not stable, right? So low energy is stability, high energy in this case is unstable, and things can really go wrong. Okay, so that is when, and I really want to just clarify this because I don't want you to think that these conformational changes, when the protein folds differently, that is actually valuable sometimes. This is not a good situation. This is when the amino acid sequence is not in the right location, let's say. So this is pathological. Okay, so it's a distinction, and that pathological outcome can arise from mutations in DNA typically, or just swapping out particular amino acids where they shouldn't be. But proteins do go through these conformational changes, right? Naturally. So they will go into that high energy state as long as they can return back to their low energy state, their native conformation, right? But if they get stuck into that wobbly like reaction, that protein will, as it says here, it might have only 10% of the normal activity, so it loses activity, functional activity, and sometimes there's a toxic gain of function. So we start with the primary structure, the amino acid. So you have the end, the amino N, end, and the carboxyl N. So the C and N regions. And then the secondary structure, right? So hydrogen bonds between amino acids at different locations, polypeptide chain, make these alpha helix or these beta or pleated sheets. Okay, and then that really gives this solid, stable structure that ends up as the tertiary structure and then allows that protein to interact with others in these quaternary structures. Prions really exist in two conformations. Most of us and most cattle and sheep and deer and elk and other mammals, mice, have a largely alpha helical shape to them. So there's I think 7 alpha helical domains in the native conformation. And it's referred to as PRPC, so I already kind of talked a little bit about this on a Wednesday. But then there's the infectious form, okay, and that largely has beta sheets. So this is just an example of secondary structures gone wrong. So you go from one that's very alpha helical, and it's not only alpha helical, there are beta sheets in the native conformation, but it's mostly made up of alpha helical, and I have a picture that'll come up, so I can just shut up and show you the picture when it comes up. But the other one, when it becomes infectious, right, those secondary structures go from alpha helical to beta sheets, make the protein thermodynamically unstable, and actually super cool, unless you have the disease. So the thing is that once you have this infectious thermodynamically unstable protein, right, so it's called PRPSC, as in it was discovered in sheep, so scrapie is the disease in sheep. Okay, that infectious protein then alters other proteins. So it takes a native conformation protein and causes it to misfold. And now you have 2 misfolded proteins. And then those go and cause other proteins in their native conformation, or the prion proteins, to misfold. And so it just keeps propagating. So it's this self-perpetuating propagation that results in pathology. Okay, so prion infection is usually, it's called spongiform encephalopathy, but it also results in these, quite striking behavioral changes. So in sheep, they start to rub up against everything and they just have this really weird sensory and motor deficits. In cows, it's the same outcome, right? They have these really crazy motor problems. And then in humans, we also have motor problems, but what the brains look like of anything, any creature that has a prion infection is it gets full of holes and it degenerates. You can't really see the holes there, but it has this spongiform-like pathology. So cells die off, and then the ventricles start to expand because there's no neurons. So Creutzfeldt-Jakob disease is the most common form of prion disease in humans, and you can get that from eating a cow or eating neural tissue from a cow that had mad cow disease or the prion disease in cows, or sheep for that matter. Okay, so those prion proteins are so similar across all these different mammals, mice included, elk, sheep, cows, humans, that they interact with each other, and once you have an infectious one, then it spreads to the brain like wildfire. It's really uplifting. Actually, the worst one is I'm trying to think of what the name of it is, fatal familial insomnia. So so in that prion disease, it's familial, so it's genetic, but it's really, really super rare. But you basically can't fall asleep for like months and you die, right? Because you can't sleep. It's an insomniac disease. It's really strange. But it's based on these prion diseases. Okay, so prion scrapie results in a disease state due to the change in the secondary structure, right? So we're talking about protein structure, not crazy diseases that affect the central nervous system. So from mostly alpha to mostly beta. Okay, so this is where you can see some of the change. So you go from the native conformation protein, right, the non-infectious PRPC that has, just on this example, 3 alpha helices. Okay, whereas the scrapie version, right, so you have a few, there's only maybe 2 in this example, but now you have 4 beta sheets, right, and here you only had 2 beta sheets. Okay, so the change in that second, so this is the purpose of this example is to talk about secondary structures, right, and how they lead to a stable protein. And in this case, the stable protein has mostly alpha helices, so I'm going to erase the beta, whereas the unstable protein, the infectious protein, has more beta sheets, has more of those secondary structures. And as it has more of those secondary structures, it becomes toxic. And so what happens is, this is just kind of a side note on all this. So I'm just going to put it up to look at and not to get too caught up on, but just for your own information. So what happens is when you have, so this would be our PRPC, right, with all the alpha helices, and then you have the PRP scrapie, or the infectious form, with beta sheets. And what will happen is that abnormal protein, that mutated protein, will start to convert other proteins into their misfolded shape. They get stuck in that misfolded shape, and then that's it. The proteins accumulate, they start to kill off neurons, and then they lead to these neurological symptoms like the motor deficits or insomnia or whatever it is. Single polypeptide chain with one or more protein secondary structures called the protein domain. So it's one single amino acid structure. Okay, so this is just really mainly to distinguish it from the quaternary structure, which is often 2 different polypeptides that come together. Okay, that don't necessarily have to be a single string. So technically, in this example, the G protein coupled receptors, right, that's a tertiary structure because it's one single amino acid sequence. I want to re-stress this idea that protein misfolding is a good thing as long as it's temporary and reversible, right? So here's one example of so remember I talked about the estrogen receptor. So it's this intracellular receptor that exists. It's the ER alpha, so the estrogen receptor alpha subtype, and it sits inside the cell. The off position, well, in fact, you can see what they're representing is that helix 12 that's in the off position, but once estrogen itself binds, then that helix 12 you can see goes into what's called the on position. Okay, so it's just a simple little fold. It's infolding. Okay, so when the estrogen receptor is bound to the ligand, it goes through this conformational change, and now it becomes active. Right, so we go from inactive to its active state, and it's higher energy. So once it's in its high energy state, right, now it can do something. Okay, so tertiary structure, native conformation, folding, but then the reverse of that. So those conformational changes and then that reversible process, that is really critical. Because otherwise it typically leads to pathology. Okay, so the quaternary structure. Okay, so now we get into basically just a bunch of tertiary structures stuck together. Maybe they want to be, maybe they don't want to be. We don't care. They just happen to be. So it's many proteins made up of multiple polypeptide chains or protein subunits. Okay, so this is now really pushing us into the next part of this course. The subunits can be the same, homodimers, or different, so hetero, tri, right? All these different dimers that can happen. You can have 2 different ones, 3 different ones, 4 different ones. Okay, so the quaternary structure refers to how these protein subunits interact and arrange themselves to form a larger protein complex or something that can really be like uber functional. Okay, and then there's also these stable interactions, hydrogen bonding, disulfide bridges, salt bridges. The GABA receptor, it's made up of different subunits. So different tertiary structures, so 1, 2, 3, 4, 5 different subunits that come together, have that hydrophilic core that allows, right, water to pass through. And then the hydrophobic surface, there are 5 tertiary structures that come together, 5 subunits that come together to make this quaternary structure. And it still is based on the same principles of unfolding, doing a job, and then refolding, going from its native rest conformation of closed to open and allowing chloride to come through.